My name is Nick Spitzer. I'm a professor of neurobiology in the Division of Biological Sciences at the University of California, San Diego. Neurotransmitter switching is a recently appreciated form of brain plasticity involving the loss of a transmitter, the gain of a transmitter, or the replacement of one transmitter with another. It occurs in response to sustained stimulation, leads to matching changes in the expression of postsynaptic receptors, and causes changes in behavior. We're fascinated by all of this. In this study, we found that sustained exposure of Xenopus larvae to odorants generated by siblings led to increased numbers of dopaminergic neurons in the accessory olfactory bulb, or AOB. Exposure of larvae to odorants generated by non-sibling larvae led to increased numbers of GABAergic neurons in the AOB. There was no neurogenesis or apoptosis, and the evidence for transmitter switching seems compelling. Identifying the right model with which to address a question was an important lesson I learned from Steve Kuffler. Davide Dulcis has an extraordinary eye for such opportunities. How did you discover this model, Davide?
I came across some early work of Waldman, and I mean very early, when dinosaurs roamed the earth, and Nick was already a full professor.
Waldman discovered that social recognition in Xenopus is mediated by waterborne kinship odorants. Based on these odorants, tadpoles show aversion towards non-siblings and attraction to siblings. Kinship odorants are processed in the vomeronasal organ by olfactory receptor neurons. The neurons project to the accessory olfactory bulb, or AOB, a center of integration of this sensory information. The AOB is composed of interneurons that can release dopamine, GABA, or both, and onto the mitral cells. Projections from the mitral cells then control swimming behavior. What happens at this level determines attraction or aversion. What we discover is that tadpoles raised in the presence of non-kin odorants, what we call the non-sibling condition, have a very different AOB. There is a sharp increase in GABAergic interneurons at the expense of dopaminergic cells, a clear sign of neurotransmitter switching. Interestingly, the switching corresponds to a marked change in behavior because these animals are now attracted also to non-siblings.
Using sophisticated pharmacology in freely behaving animals, Davide discovered that the two different classes of interneurons control opposite behaviors in normal animals.
We implanted agarose beads loaded with drugs just below the AOB and tested the role of the two transmitters. We found that dopamine is crucial for attraction and GABA for aversion, suggesting that the profound neurotransmitter switching occurring in non-siblings is causing the changes in social preference. But what coordinates neurotransmitter switching at the molecular level? And here is where the Italian connection came to the rescue. Giordano, a project scientist in Darwin Berg's lab next door, is an expert in microRNAs, and it took only a few glasses of wine to convince.
A few bottles, you mean? MicroRNAs were ideal candidates to mediate neurotransmitter switching. MicroRNAs are post-transcriptional repressors of gene expression. A single microRNA can target many different messenger RNAs to consolidate specific genetic programs and trigger developmental switches. It just made perfect sense. So we microdissected accessory olfactory bulbs and sent them for small RNA sequencing. We found that several microRNAs were upregulated in non-sibling conditions versus sibling conditions, and two in particular had key roles, microRNA 375 and microRNA 200B. Both these microRNA are targeting key factors that determine neurotransmitter identity. One of them is PAX6. If the level of PAX6 are high, the neuron becomes dopaminergic. Increase in microRNA 375 reduces PAX6 levels and changes neurotransmitter identity. This explains why, compared to sibling condition, where we have many cells that express dopamine alone or in combination with GABA, we see a drastic drop in dopaminergic cell in non-sibling condition. The second factor is BCL11B, a known inhibitor of the pathway that induces neurons to become GABAergic. Increase in microRNA 200B removes the inhibitor, explaining the drastic increase in GABAergic cells that we observed.
The mechanism for switching social preference in response to olfactory stimuli may be relevant to other species. One thinks of olfactory imprinting of zebrafish and salmon, neonatal attachment in both mice and lambs, and potentially of people.